Question number 12. Photosynthetic bacteria have pigments in. The given options are chromatophores, B. leucoplast, C. chromoplast, and D. chloroplast. So here the bacteria are prokaryotic organisms. So prokaryotic means the one which does not have the double membrane structures like plastids and mitochondria. So here the plastids are absent. Here plastids are of three types. Those are called as leucoplast, chromoplast, and chloroplast. Actually, to say, leucoplasts are the colorless plastids which are concerned with storage of food materials, and chromoplasts are the colored plastids. The one which is providing all colors except the green color, and chloroplasts are the green colored plastids. Here these green color plastids are the one generally will contains the green colored pigment called as chlorophyll pigment. Like as we know that chlorophyll A, B, and are the main chlorophyll pigments which are responsible for the photosynthetic process. These are the one which are present only in the eukaryotic organisms, but here as we are saying bacteria is an prokaryotic organism, it will lack all these structures. But when we consider that photosynthetic bacteria are present, that means which contains the chlorophyll molecule. Of course here they does not have the normal chlorophyll as like that of eukaryotic organisms, but they have a special type of chlorophylls called as bacterial chlorophylls. These bacterial chlorophylls are present in chloroplast like structures, but exactly they are not the chloroplasts. Those structures are called as chromatophores. Chromatophores are the structures in which the pigments, that is photosynthetic pigments, are present. So from the given options, option A is the correct answer. Question number 13. Bacterial cell membrane has, the given options are A. chitin, B. cellulose, C. proteins and phospholipids, and D. acetyl glucosamine and muramic acid. So here this is a question related to cell membrane or plasma membrane. Cell membrane or plasma membrane is a living membrane which is present just below the cell wall, and here the given options like chitin. Chitin is the cell wall material in fungi, which actually chitin is also a type of polysaccharide molecule, but it is a nitrogen containing polysaccharide. And second one, cellulose. Generally cellulose is the one which is considered to be the cell wall material of plant cells. And third one, proteins and phospholipids. Actually if you take any living organism, the plasma membrane, the basic composition that is present in the plasma membrane is proteins and lipids, and of course the lipids contain phosphate molecules, so we call them as a phospholipids. As bacteria is also a living organism, it also contains the plasma membrane just below the cell wall. That plasma membrane is made up of the proteins and phospholipids as like that of other living organisms. And last one that is given as acetyl glucosamine and muramic acid. So, acetyl glucosamine, muramic acid are the cell wall materials in bacteria. So, chitin is a cell wall material in fungi, cellulose is a cell wall material in plants, and acetyl glucosamine and muramic acid are the cell wall materials in bacteria, whereas this is a plasma membrane material or cell membrane material present in all living organisms including the bacteria. So, from the given options, option C is the correct answer. Question number 14. Cocci are A. Ciliated, B. Flagellated, C. Non-ciliated, and D. Both 1 and 3. So, cocci are the spherical shaped bacteria. Here all cocci are non-motile in their nature. So, actually when we say it as motile, they should contain either or other type of either one or the other type of locomotor structures like a flagella or cilia. With the help of the flagella or cilia, they will move in the water or whatever the environment in which they are existing. But here all cocci are non-motile, that means they does not have any special organ for locomotion. That means neither they are ciliated nor flagellated. That means they does not have cilia, and at the same time they does not have flagella also. So, here from the given options we can say that cocci are the non-ciliated. So, from the given options, option C is the correct answer. Question number 15. The method of genetic recombination or parasexuality in bacteria is known as. Given options are A. Conjugation, B. Transformation, C. Transduction, and D. All of these. Here, if you consider the sexual process in bacteria, generally, before considering the bacteria, when we consider it as a sexual reproduction, there should be gamete formation and the fusion of the gametes and formation of the zygote leading to the further divisions, like it may be either by mitosis or by meiotic process. But here in bacteria, no gamete formation. And no gamete fusion, and there is no zygote formation. So, whatever the characters where we consider it as a sexual process, those characters are not exhibited by the bacteria. That means sexual process, the real type of sexual process is not existing in bacteria. But when a sexual process is taking place, the main thing is the transfer of genetical material is present. But here, the same time, genetical recombinations are also possible during the sexual process. In bacteria, these three things are not present, but the thing is the genetic recombination will be taking place, that we call here it is not by the gametic fusion, gamete formation, so we call it as a parasexuality, and here it is done by three methods. One is called as conjugation, transformation, and transduction. That is, in these methods, like one process is where two bacteria will come and attach with each other with the help of that sex pili, or we can consider it as a through the conjugation tube, the genetic material will be transmitted from one bacteria to the other bacteria. We can call it as a direct contact of bacteria. The genetic material will be transferred from one to the other. And there is one more process where the bacteria which is present in environment will burst and release all that genetical material, or the genetic material which is present in bacteria will be broken into small fragments. And if a broken fragment is get attached to any other bacteria, it will be directly absorbed through the cell wall of that bacteria. That is one method called as transformation. And in the transduction process, where the bacteriophages will help in the transfer of genetic material from one bacteria to the other bacteria. So in all of them, the genetical material is transferred from one bacteria to other bacteria, leading to the genetical recombination process. So here all the three methods are being the part of the genetic recombinations in bacteria, that is conjugation, transformation, and also the transduction process. So all of these, that is option D, is the correct answer. Question number 16. Viruses which infect bacteria multiply and causes their lysis are. The given options are A. Saprophytic, B. Temperate, C. Avirulent, and D. Virulent. So here it is the one which is related to viruses. Viruses will infect the bacteria in the process of lytic and also the lysogenic cycle. So when they, that is in the life cycle of viruses, or we can say that a sexual process of viruses, the viruses will infect the bacteria, and when they infect the bacteria, with the help of an lysozyme enzyme, they will form an pore on the cell wall of a bacteria. Through that pore, the genetic material of the bacteriophage or the viruses will be injected into the bacterial cell. When the injected DNA part of the viruses will get incorporated into the the bacterial DNA, or sometimes it may help in the further process, it may help in the lysis of the bacteria. So here in this one, it is the given options that have been given as a saprophytic. Saprophytic means it's a type of nutrition where the parasitic organism, or we can say that the dependent, will absorb the nutrition from the dead and decay organic materials. So that is not correct related to this one. Second one is temperate phases. Temperate phases means the temperate phase of the bacteria, where when the viral DNA is incorporated into the bacterial cell, the viral DNA will get fused with the bacterial DNA and converted into the prophase part. So that is not causing any harm to the bacterial cell. Such type of viruses are called as temperate phases, and these are participating in the lysogenic cycle. That is, consider this as a bacteria, and this is a bacterial DNA part. This is a viral DNA that have been incorporated into the bacterial cell, and this viral DNA it is drawn as a thick one, so to differentiate from here, so this will get incorporated into this bacterial DNA, and it appears to be like this, where the thick part will be shown as a viral DNA, this part, and remaining part will be the bacterial DNA. This is called as prophase, and when this bacteria will undergo binary fission, the DNA material will be transmitted to the bacteria equally, and such type of phases are called as the temperate phases, which will not cause any harm to the host bacteria. And third option is given as avirulent. Avirulent means if it is not causing any harm, so we call them as a avirulent phases. But here in the process of lytic cycle, the viruses are making the bacteria to burst or undergo lysis, so it is not avirulent. And the last one is virulent or the virulent one. Here in the virulent or virulent one, where it will be participating in the lytic cycle, where in the last step of lytic cycle, the bacterial host will burst and release the viral particles. So that is leading to the lysis of the bacterial cell, and that is causing harm to the bacteria. So we call such bacteriophages or the viruses as virulent bacteria. So from the given options, option D is the correct answer. Question number 17. Which one of the following statements about viruses is correct? The given options are: A. Viruses possess their own metabolic system. B. All viruses contain both RNA and DNA. C. Viruses are obligate parasites. And D. Nucleic acid of viruses is known as capsid. So here it is the one which is related to viruses. So here actually viruses lack their own metabolic process. So here, but it is not like that they does not perform metabolic process, but they perform this metabolic process. They does not have their own metabolic system, but when they attack an host, they will use the metabolic system of the host for their functions. And generally the viruses have genetic material. The thing is, either the genetic material will be in the form of RNA or DNA. That means no viruses will have both type of genetic material within the same virus. And viruses are obligate parasites. That means parasites are the one which will depend upon the living host for their nutrition. And here without the living host they cannot survive. That's the reason why these are considered as the obligate parasites. And if you consider the structure of the viruses, viruses will contain the protein part. The protein part is called as capsid, and inside which the nucleic acid will be part. So this part is called as a core region. So here the nucleic acid part is the central part which is considered to be present in the core part, and surrounding that nucleic acid part the capsid, that is protein part, will be present. So here from the given options, obligate parasites, that is viruses cannot survive in absence of the living host, is a correct option. So from the given options, option C is the correct answer. Question number 18. Viral genome incorporated and integrated with the bacterial genome is referred as. The given options are: A. Prophase, B. RNA, C. DNA, and D. both 1 and 3. So here during the lysogenic cycle in viruses, what happens is the viral particle will attack the bacteria. When it will attack the bacteria, it will release the viral genetic material into bacterial cell. When it is released, okay, actually here before that we should know that how it will be released means the viral particle will secrete some enzyme called as lysosome, which is an cell wall digesting enzyme. It have the ability to digest the cell wall of bacteria, that is called as lysozyme, and it forms a perforation on the cell wall of bacteria. Through that perforation, the viral genetic material will be injected into the bacterial cell, and here this is a bacterial DNA and this is a viral DNA that have been incorporated here. So this will get incorporated into the bacterial DNA, and now this structure where this is a viral DNA particle. So this is a bacterial DNA or bacterial genetic material part, and this one is a viral genetical part. And here Now it is a combination of both type of genetic material, and this structure is called as prophase. So when viral genome is incorporated and integrated with the bacterial genome, the structure is called as prophase. So from the given options, option A is the correct answer. Question number 19. Bacteriophage release lysozyme during. Given options are A. Penetration phase, B. Eclipse phase, C. Absorption phase, and D. Maturation phase. Here what happens is when the bacteria is attacked by any viral particle like a bacteriophage, this is a bacteriophage which have been attacking this bacterial DNA. Generally the bacteriophage, so this is a bacteriophage. This is a host bacteria. Generally here what happens is the bacteriophage will secrete an enzyme called as lysozyme enzyme. Before this, one thing we have to know that in the lytic cycle, the first step is the collision between the bacterial cell and the bacteriophage. That means the bacteriophage will identify some specific sites on the bacterial surface, and it will attach to that particular specific site only with the help of the tail fibers. When it attached, that is called as absorption phase, and after absorption, so this bacteriophage will secrete an enzyme called as lysozyme, and that lysozyme will form an perforation on the wall of the bacterial cell, and that will occur during the phase called as penetration phase. That is during the penetration phase, the bacteriophage will secrete that enzyme called as lysozyme, which will digest the bacterial cell wall. So from the given options, option A is the correct answer. Question number 20. Viruses as contagium vivum fluidum was said by. The given options are A. Mayer, B. Stanley, C. Beijerinck, and D. Ivanowski. So here viruses were when they were initially discovered, so they were known by different concepts, because when they have been first discovered, they come to know that they are passing through the bacterial filters, and some they consider it as they are poisonous, or they are causing some site of toxic nature to the living organism. And initially they were discovered, they come to know that it is not, and it is having some sort of fluid like nature. So it is contagious and causing some damage to the living organisms, and it is having the fluid nature, that is called as contagium vivum fluidum. Of course, later the concept was discarded because the virus are not having the fluid nature, they are having the particle nature. So this concept of contagium vivum fluidum was given by Beijerinck scientist. So from the given options, option C is the correct answer. Question number 21. Virion is. The given options are A. Nucleic acid of virus, B. Antiviral agent, C. Protein of virus, and D. Complete assembled virus. So here virion is nothing but an viral particle. It is single viral particle we can consider, and we know that viruses are the nucleoproteins. So nucleoproteins means, nucleo means they are made up of nucleic acids, or they contain nucleic acids, and along with the nucleic acids they are having proteins also. So here Virion is a complete assembled viral particle which contains both the nucleic acid part and also the protein part. So here from the given options, option D is the correct answer. Question number 22. Viruses were first crystallized and isolated by. The given options are A. Ivanovsky, B. Borden, C. W.M. Stanley and D. Smith. So here initially the viruses were discovered. They were known by different concepts that they are having fluid nature and they are having nucleoprotein, all that characters they used to be said. But when the scientist, that is W.M. Stanley, he had first time crystallized that viral particle, that is when it have been crystallized, it is easy to observe and also study the nature of these viral particles, and he had separated, that is isolated and crystallized. So the first viruses which have been crystallized was the TMV virus, that is from the cultures of tobacco or infected tobacco leaves, that viral particle was separated and it have been crystallized to know the nature of the virus particle, and based on that one he said that they have the nucleoprotein nature. So from the given options, option C is the correct answer. Question number 23. Viride have. The given options are single stranded RNA not enclosed by protein coat, B. single stranded DNA enclosed by protein coat, C. double stranded DNA enclosed by protein coat, and D. double stranded RNA enclosed by protein coat. So here viroid. Viroid is a viral particle which is made up of only nucleic acid. So and this is a structure which lack protein or capsid, and here this nucleic acid may be either made up of RNA or DNA structure. That means viroid is a structure which is made up of the nucleic acid without an protein coat. As I said that the nucleic acid may be RNA or DNA. So here from the given options we can say viroid have single stranded RNA not enclosed by protein coat. So from the given options, option A is the correct answer. Question number 24. The protein coat of virus is called. The given options are A. capsid, B. cosmid, C. capsomere and D. chromosphere. So here the viruses are having two structures. One is called as a nucleic acid part and the other part is called as protein part. The protein part is called as capsid, and the units of capsid, that is the individual units of capsid, are called as capsomeres, and the protein part is acting as a protective sheet around the central core of nucleic acid. So here the protein coat of viruses is called as capsid. So from the given options, option A is the correct answer. Question number 25. Viruses are. The given options are A. cellular organisms, B. non-cellular organisms, C. unicellular organisms and D. cellular without wall. So here viruses are the organisms which are having some specific characters, that is these are called as obligate parasites, and these are the one which does not have cellular organization, or we can say these are acellular organisms. So here when we are calling it as acellular organisms, they lack cellular structure. Of course, when they lack cellular structures, how could we consider them as a living organisms? Means they are the one which are made up of the genetic material, that is they have their own genetic material, as none of the non-living organisms have their own genetic material. Based on that character, we can consider the viruses as a living organisms. But the thing is, they does not have cell wall, they does not have plasma membrane which will encircle that genetic material. So those are the specific and unique characteristic features which can be seen in the viral particles or the viruses. So from the given options, we can say that these are the non-cellular organisms. So from the given options, option B is the correct answer. Question number 26. Viruses are essentially made up of. The given options are A. Proteins and nucleic acid, B. Proteins and carbohydrates, C. Lipids and nucleic acids, D. Starch, proteins and lipids. So here, when you see the nature of the viruses, viruses are generally called as nucleoproteins. That is, they are made up of nucleic acid either in the form of RNA or DNA molecule, and second part is a protein part which is acting as an protective sheet around the central core of nucleic acid. This part is called as a capsid. This is a general organization that can be seen in the viral particles which are made up of the proteins and the nucleic acids. But exceptionally, there are some viruses which contains carbohydrates also along with the nucleic acid part and the protein part. But in general, majority of the viruses will have proteins and nucleic acid in their composition. So from the given options, option A is the correct answer. Question number 27. The TMV capsid has number of capsomeres. The given options: A. 2130, B. 1230, C. 3210 and D. 2310. So here the TMV virus, which is considered to be the one of the typical virus for plant viruses, and this is the one which was first crystallized by W.M. Stanley scientist. And here if you observe the TMV virus, so it will have a central core. This central core is made up of nucleic acid like this. The nucleic acid part is also in the form of a helical structure like this, and around this helical structure we can see the protective structure which is made up of the protein subunits. These protein subunits are generally called as the capsomeres like this. This is a structure which we can see in the TMV viruses like this. And if you observe, this is having a nearly a rod shaped structure. So this is a RNA molecule, that is a single stranded RNA is present in the center, which is considered to be the core part, and around that one, so this entire part is called as a capsid actually, and each unit is called as capsomere. Individual units of capsid are called as capsomeres, and the capsid, as we know, it is a protein part, and the total number of capsomeres which are present in the capsid of TMV is 2130. So from the given options, option A is the correct answer. Question number 28. A virus can be considered as living entity because it, the given options: A. reproduces inside the host, B. can cause disease, C. responds to touch stimulus, and D. shows metabolism. So here the viruses are the one which will participate in the process called as reproduction. Yes, but here it is not outside the host, it is always inside the host, and of course this is one of the reason why we can consider this viruses as a living organisms. And one more thing, of course, they will cause diseases also. It is not compulsory that because of this specific reason they should be considered as a living organisms. And of course, they does not have response to the stimulus, response to touch stimulus, and shows metabolism. Of course, they does not have their special individual metabolic system. That's why this is not been related to the viruses. So from the given options, because as they participate in the reproduction inside the host, because of this reason we can consider the viruses as a living entity. So from the given options, option A is the correct answer.